And to discuss with you about the role of AI for radiomics analysis, first of all, we need to discuss a bit about what radiomics is as a type of analysis. And then I will give you a flavor on how AI can be used to augment this type of analysis, and you'll see that they go together in most of the times. And of course, what are the challenges and the clinical, in the clinical implementation of radiomics types of analysis, and of course, how we can use AI to alleviate those problems, overcome those challenges in the end. So everything started at this time in 2014, where this paper came out to basically show exactly what we discussed in the previous talk, that we can have information, we can extract information from images that cannot be seen with the human eye. And this information can give us a lot of detail about the phenotype and the biological background of tumors, for example. And, oops. Can we have some assistance? Thank you. Okay. So, as the title of this editorial in Radiology says, radiomics are the way that we can get data from images. So images are more than pictures that we see, they're data. And this started a whole series of publications that is now skyrocketing on radiomics. And to understand what exactly radiomics is, I would like to take you back to basic biology. Apologies to the friends in radiology that have been away from that, and I had been away from that for a while. But we need to start thinking about the cell. And the cell starts with information from the genes that are transcribed, translated later onto proteins, and basically expressed as the phenotype of the cell. So genes, mRNAs, proteins, metabolites, all these things within the cell compile a function that will give us a cellular phenotype. So nowadays, we have methods to extract information from all those molecules, from mRNAs, from proteins, from metabolites. We can get a series of tests that can give us detail on all the metabolites, all the proteins, and all the mRNAs that we can get from one cell, for example, or from a series of cells, a collection of cells. In the same way, we can have information about tissues, information about organs by extracting a lot of detail, textural information, and other types of information from images. So this is what you see in a radiological image, but in the background, you can find information that can be used to give us an idea about the phenotype and the actual function of the tissue. So in the scale of omics analysis, as you see in this graph, we start with a smaller scale in genomics, we go to transcriptomics, proteomics, metabolomics, and as the scale increases, at the top level, we can find radiomics, which is actually what we see in a macro level using the radiological techniques that we have now available. But this graph gives you the idea that all this information that we get from radiomics is actually information that is reflects what happens in the lower scale. So something that happens at the proteomics level will actually impact what we see in our radiological image later on. So combining these types of information can also give us some nice details about the function of the tissues. So think of radiomics as the upper scale, the larger scale, from what we see, that reflects the function of the tissues and what we have in the lower scale, for example, the function of our genome, the mRNA, the proteins that we have in our cells. So now that we understand what exactly radiomics is, I would like to show you the two types of radiomics that we have. And the two types have to do with the way that we extract the data from the images. First of all, we can get radiomics data by extracting features manually, selecting the region of interest on an image, and then predefining a series of mathematical functions that will give us a detail, that will give us basically numbers, a series of numbers from that region of interest. That series of numbers is the radiomics features that we call handcrafted radiomics features, just because that we have predefined a series of functions to derive those numbers. When we have that set of numbers, we can use it for machine learning, and we can do all sorts of things that you've seen in several papers that we have in literature at the moment. And of course, you can do this in several ways, but there have been initiatives to standardize the way we extract handcrafted radiomics features, and the most important one is the Image Biomarker Standardization Initiative, the IBSI, as you've seen it in literature. And all the methods that we actually use at the moment, most of them are compliant with the guidelines set by the Image Biomarker Standardization Initiative. And the most important one that you will see in 90% of the papers out there is PyRadiomics. PyRadiomics is basically a set of tools which is IBSI compliant, complies with the guidelines set by the IBSI, and gives us the ability with Python, with coding, to extract features that can be extracted using open access software, for example, 3D Slicer. And there is extensive documentation on how PyRadiomics extracts those features, and this is a standardized method. You can go online, you can find a list of all those features, and you can have an idea of how they're calculated. So every person in the world that uses PyRadiomics extracts features with the exact same way as everyone else. The second way to extract radiomics features is using a deep radiomics pipeline, and this means that we actually, instead of defining functions, mathematical functions to get numbers from the images, we actually use neural networks that decide how these numbers are extracted. So this is automated, and this is not predefined. So in a sense, it's a kind of a black box application, but has the ability to give us very, very detailed information. So this is an active field of research, and deep radiomics will get into literature more and more within the following years. Of course, the values that you extract with deep radiomics, as the values that are extracted with handcrafted radiomics analysis, can be also used for machine learning analysis at the end. So you'll see in most of the papers that radiomics and AI go together. They dance together, as you'll see in the images, and the result comes from a combination of a series of methods. And to understand how AI can augment, basically, this radiomics workflow, you can see that the radiomics workflow starts by getting the images, first of all, pre-processing the images, preparing them for analysis, extracting the regions of interest for the extraction of radiomics features, the extraction of features itself, which is basically handcrafted or deep radiomics, as we discussed, selecting the ones that are suitable for model development, and finally developing a machine learning model. At all these steps, AI algorithms can help remove problems that we have in the analysis, and of course augment and automate the analysis. And I'll give you some examples. First of all, as you already saw in the previous presentation, we can have AI generate images, and the biggest problem that we have for those types of analysis, for radiomics analysis specifically, is the low sample size and the small samples of images that we have for a specific disease, because of disease prevalence, because of the low number of patients that we had in our department. So we can use AI to synthesize images from source images, and this is an example from images that we have synthesized in our department. So you see the intermediate weighted fat suppressed images of sagittal of the knee on the left. On the right, far right, you can see the conventional gradient echo images acquired with our machine. But in the middle column has synthesized images, synthetic images that have been generated with AI algorithms. In this way, you can actually get realistic looking images that can be used for radiomics analysis later on. And this is another example as the one you saw previously with 0.55 Tesla machines, where you can use super resolution to enhance what you see on the image and make the image smoother and nicer to diagnose. But be careful, because as you see, going from the left, which is the conventional image, to the right, the far right, which is enhancing more and more using deep learning algorithms, you'll see that on the far right, you lose information about the texture of the bone, you lose information about the trabeculation of the bone there. So you need to be very careful when applying those algorithms, because if you overdo it, you may lose important information. You could potentially lose a hairline fracture there. Battery was running low. Let's give me a second. And then, if you can create new images, you can also... We need a charger. Laptop has run out of battery. Is it this one? Okay, so if you can create synthetic images, you can solve problems that we have in radiomics analysis. One of those problems is representation bias. So if you try to predict what happens in a disease that has low prevalence, and you train your data using different prevalence in the training set compared to the prevalence of the disease in the test set, you may end up having fake predictions that are not correct at the end of the day when you try real-life applications of those models. So you can use models, AI models, to enhance, to create representations in the data set that are close to the real population, so that you can solve problems like this and have radiomics analysis that gives representative results at the end of the day. And of course, if you have those types of images enhanced by deep learning, you can see that the information you can extract is also similar to the one that you can see in the images. And with 2 to 5 minutes acquisition time, for example, for an E protocol, you can get information that is diagnostically equivalent to the information that you see with a bare eye in the conventional long protocols that we have. But we need to think, can we use this information in radiomics analysis? So it is interchangeable with regards to the human eye, but is it interchangeable in terms of texture? This needs to be studied. And then we need to select the region of interest to extract the information, and we have models as the models that you saw for the spine earlier on that can automatically detect structures across the body. These are some of the models that you can find out there, and these have been used to do large-scale segmentation within the body, and of course with any type of tissue that you can imagine. And this is the total segmentator model that has been incorporated in several types of open access software, for example 3D Slicer. And with the total segmentator, you can extract segments from CT and from MR. MR is being released soon. A new version of it is being released soon. And you can see that by segmenting all these tissues, you can do large-scale radiomics extraction. And we've done this. We've used the total segmentator in our group to extract information from 50 tissues from 500 patients, something that doing in the past by hand would take ages to finish and would be a very, very, very tedious and laborious task to complete. So being able to automatically segment so many tissues at one go, it's an important advantage, especially when you try to do whole organ segmentation and whole organ radiomics analysis. And these models can be coupled, the segmentation models can be coupled with neural networks that can be used to do deep radiomics analysis. And this is why deep radiomics analysis is useful, because it can be coupled also with other methods and do automatic extraction of radiomics features. The problem there is interpretability, because if you do handcrafted radiomics, you have a pretty defined set of algorithms that have been used to extract the numbers. If you use convolutional neural networks, for example, to extract deep radiomics features, you don't know how the network has extracted the features to end up in creating the models. These features can be selected, can be reduced in number to be used later in models, and AI algorithms like Boruta feature selection can be used to tell us which ones are relevant and which ones are not. Of course, you cannot create a model that has 1,000 features in there, so you need to reduce the number and take 10, 20 more relevant features to create your models and create something that will work actually later on. And finally, what you see, the tip of the iceberg that you see in publications with radiomics is the development of models. And of course, you have various architectures, starting from leader models and smaller traditional architectures like SVM. And of course, you can use even neural networks and deep learning to analyze and create more complex models with radiomics. These are not usual, these are not very common, because complex deep learning models are an overkill, let's put it this way, are an overkill to analyze radiomics data. But at the end of the day, when you have your model, you can actually use algorithms like clustering analysis to understand the structure of your data, to explain what happens. And you see here that you can see very pronounced differences between the groups in a disease that the radiomics predicted which people would die and which people would be alive after some treatment. And radiomics can be explained and can give you information about this thing as well. But once you have the model, you can also develop what we call subgraphs, supply explanation graphs, where you see the contribution of radiomics features and how much each of the features has contributed to the final result of the model, to the final prediction. So overall, you can see that we can have AI algorithms that can be used at all steps of radiomics analysis and can give you information and can enhance the analysis so that you solve problems that come along the way. So in the data availability part, for example, you can have generation of synthetic images. In the representation problem, you can have enhanced sampling and increase of the lower represented groups, for example. You can explain what radiomics show using class activation heat maps and SAP values. And of course, with AI-enhanced image acquisition, you can increase the reproducibility and the quality of the data that you put in the algorithms so that you can have nice models predicting what you want to predict. If you want to apply those in your practice, I suggest you refer to our recent guidelines from the European Society of Medical Imaging Informatics, where you can find how to do radiomics analysis, the description, but also code. So if you are keen to code yourself, you can code there, you can find the code ready for application, and you can start doing your own radiomics analysis. And of course, all the things that I have described can be found in this recent publication from our group. Thank you very much.